Precision medicine for pancreatic cancer, targeting the DNA damage response. Rather than a one-size-fits-all approach, precision medicine aims to deliver a tailored approach to therapy, where the treatment is determined by individual tumour characteristics. A good example of precision medicine in practice is the use of BRCA mutations to select for patients with ovarian or breast cancer who will respond well to PARP inhibitor treatment, such as olaparib. It's well documented that cancer cells which are BRCA mutant are hypersensitive to PARP inhibitors such as olaparib, and these preclinical findings are now translated into clinical benefit, such as in the SOLO-1 trial. This is true not only in ovarian and breast, but also in pancreatic ductal adenocarcinoma, as evidenced in the POLO trial, and even more recently in prostate cancer patients as well. Given the success of using BRCA as a predictive biomarker for PARP inhibitor treatment, there's great interest in the field to get this model to work for other targets. Today I'm going to talk about pancreatic cancer, and a therapy I'm going to talk about is a combination therapy called gemcitabine combined with an ATRi inhibition. The key question being what would be a good biomarker for this therapy and whether or not ATM could fill this gap. So pancreatic ductal adenoma is a disease with very poor prognosis, and it's characterized by mutations commonly in DNA repair genes such as ATM. ATM is a master regulator of cellular responses to double-strand breaks, as well as being directly involved in repair processes such as homology-directed repair. It also can induce cellular events such as cell cycle arrest or apoptosis. ATM is commonly mutated in a proportion of pancreatic cancer patients. Germline alterations can be identified, as well as somatic ATM gene alterations in PDAC. A recent review, which captured over 5,000 PDAC patients, estimated that total overall prevalence of ATM mutations, either germline or somatic, is around 6.4%. This is at the DNA level. Tissue microarray datasets using IHC estimate that low ATM protein expression can occur in around 12% of PDAC patients, though there's no specific definition on how to define ATM low. So, knowing that ATM is seemingly defective in a proportion of PDAC, and knowing that it's a master regulator of responses to double-strand breaks, an interesting question in the field is whether loss of ATM will sensitize to treatments that induce double-strand breaks. I'm going to talk about one of these treatments, this combination here. We showed in 2018 that the ATRi inhibitor AZD6738 synergizes with the chemotherapy agent gemcitabine. So gemcitabine, as a nucleoside analog, poisons the replication machinery through multiple mechanisms, inducing fork stalling. The primary response to these stalled forks is ATR. However, if ATR is inhibited by AZD6738, the stalled forks persist and collapse into double-strand breaks, which are highly toxic. In turn, we demonstrated that this combination can induce tumour regression and is better than gemcitabine alone in multiple PDAC models. Off the back of that 2018 paper, a phase 1 clinical trial has been launched to assess this novel combination in patients, including PDAC patients. However, the question still remains of whether a predictive biomarker can be validated, and specifically whether ATM loss sensitizes to this combination. To address this question, we first simply inhibited both kinases with an ATR inhibitor and an ATM inhibitor, and found that in every PDAC cell line tested, ATM inhibition did sensitize to ATR inhibition, and this combination of the two was synergistic, as demonstrated in these benefit synergy plots, the blue areas noting synergistic interaction between the two agents. In colony forming assays, ATM inhibition strikingly sensitized to ATR inhibition. We then targeted ATM through a different method, namely siRNA knockdown, and interestingly, despite achieving quite durable knockdown of around 90%, knockdown did not sensitize to ATR inhibition, unlike the previous experiments where we're using an ATM inhibitor. This perhaps suggests that just having low ATM protein isn't enough to sensitize. So to try and explore this further, we made some ATM knockouts. We generated 3 different clones of MIA-PACA2 ATM knockouts, and these clones were 7-fold more sensitive to ATR inhibition compared to ATM positive clones. So strikingly different result to the knockdown, again suggesting that you need complete loss of ATM function to sensitize to AZD6738 in these PDAC cell lines. So what about with the ATRi and gemcitabine combination? Well, when we pulsed the combination for just 24 hours and then released into normal medium, ATM wild-type MIA-PACA2 cells were able to grow just fine, whereas ATM knockouts cells were completely flatlined at many concentrations, demonstrating significant sensitization to this combo. And this came through also in the synergy data. We found that while you need higher concentrations of both compounds to see synergy in the wild-type line and the knockout line, you can see synergy even at low concentration of the ATR inhibitor and gemcitabine. We then did some cell cycle profiling and found that specifically in the ATM knockout cells, gemcitabine induced intra-S phase arrest, which was augmented by the addition of the ATR inhibitor, again specific to the ATM knockout cells. This coincided with an increase in replication catastrophe markers, namely cells which are pan-nuclear for gamma-H2AX and phospho-RPA, serin 4/8. Again, this replication catastrophe phenotype being induced much more effectively in the ATM knockout compared to the wild type, suggesting that ATM protects against this widespread fork collapse induced by the combination. We then did some in vivo experiments, whereas the ATR inhibitor at 50 mg per kg had no effect in the MIA-PACA2 wild-type xenografts. We induced a 61% tumour growth inhibition in the ATM knockout xenografts. We then did experiments with the ATR inhibitor and the gemcitabine. The combination of ATRi gem induced growth delay in the MIA-PACA2 wild-type xenografts, but actually induced modest regression in the ATM knockout xenografts, as shown also in these waterfall plots below. So, what we believe is happening is that when you give gemcitabine in these PDAC cells and inhibit ATR, the stalled forks collapse into double-strand breaks in S-phase cells. If ATM is proficient, a lot of these double-strand breaks in S-phase can be repaired. Replication resumes, you see a normal cell cycle profile, and the cell survives with very few DNA damage markers. If ATM is absent, however, these double-strand breaks in S-phase go unrepaired. They accumulate, resulting in massive replication catastrophe. You see this pan-nuclear DNA damage and a large intra-S accumulation. So in summary, ATM loss of function for either kinase inhibition or genetic deletion sensitizes PDAC cells to ATR inhibition and to the ATRi gemcitabine combo. This sensitization is associated with an augmentation in replication catastrophe. Therefore, ATM loss of function could be a predictive biomarker response for the ATRi gem. As I've mentioned, this combination is being assessed in a phase 1 trial. Due to the important discovery that just low ATM expression for siRNA knockdown wasn't enough to sensitize, we believe it's important to get a very thorough assessment of ATM status in PDAC patients, because the distinction between ATM low and ATM null could be critical in determining response. Therefore, as part of the ATRi trial, we incorporated a multimodal assessment of ATM via sequencing, IHC, and pre- and post-treatment biopsies of HAPC DDR activation post-treatment. This work was performed at the Cancer Research UK Cambridge Institute as part of the University of Cambridge in the Duncan Jodrell group. Thanks to all group members and also the core facilities in the Institute, as well as collaborators in AstraZeneca and the funders Pancreatic Cancer UK and CR UK.